Yes, so in the last portion, we have discussed about the transcription, if you remember that. And today we are going to start the genetic code. Okay, so in the last portion, I guess you have seen that if we have the DNA, okay, this is our DNA like this. So if this is our DNA, so from the DNA, of course, mRNA is coming. So if the mRNA is coming, so both the strand help in the formation of this one or only the one strand?
Only one strand.
Yes. So if only one strand is helping in the formation of this mRNA, so both the
Proteins can be
strand
formed,
are work as
and
a
the tRNA
template
can
strand
be.
or only the one?
Say it again.
Yeah, both the strand work as a template or only one work as a template DNA?
One.
One. Okay, another is known as a coding.
Hold on,
Other
just give me
strand.
1 second. Your voice is very quiet.
Hello. Now is it fine to you?
Yeah, it's very quiet. Maybe my problem. Yeah.
So from the DNA, mRNA we have just formed, and you know it very well. Now our main purpose is to form the protein chain from this mRNA. Now it is our main role. So we want to form the protein chain from this mRNA. So before I proceed with the formation of protein chain, you should know what is the genetic code. So see, if this mRNA is getting formed, so of course it has some genetic code now because it is coming out from the DNA. Suppose it has a code like G A U U A C C G. This what? It is a genetic code.
Yeah.
Okay. So genetic code is so just tell me your mom, your dad, and you. Your genetic code on the mRNA or in the DNA, it is almost similar or different.
Similar.
But there is a condition. Your mom
Mm.
DNA and your dad DNA is different
Yes.
because they are from different family. But
Mhm.
now, your DNA, your brother DNA, your sister DNA, it is almost same because you all are from the same family, right? So that's why.
Mhm.
So genetic code means, can we say that it's a relationship between the sequence of the nucleotide on the mRNA
Yes.
sequence?
Yeah.
Okay. So it is showing the relation. So I can write it there. So genetic code is what? It is actually it is showing the relationship between the sequence of nucleotides on mRNA and the sequence of amino acid in the polypeptide.
Mm.
So it means in the DNA, how new DNA is getting formed, which type of coding should come, which type of new DNA. So it is decided by genetic code. Okay, and how you will see very soon. See,
Yep.
there was scientist whose name was George Gamow. George Gamow. So George Gamow was saying that, guys, see very carefully. We have total 20 amino acids. We want to make total 20 amino acid. Okay, in our body 20 amino acid are there by which you can form your body protein. But we have only four type of code. Like we have only four type of code for the mRNA: guanine, adenine, cytosine, uracil. Thymine is not there for the mRNA. So four type code, single code. So four type single code, only code for the four amino acid, only code for four amino acid, but we need 20 amino acid. If four code for four amino acid, so how 16 amino acid would get formed? We need the code for other 16 amino acid also. So
Yeah.
finally George Gamow said that these are present. There are
Mhm.
four main code, but actually they are not present single. It means if these code help in the formation of amino acid, so it is not single. It is not individual. Maybe they are connected together like doublet. Okay, maybe like this: Ga, Cu, Ca, Gu. So can you tell me how many combination will get formed from these four if you
16.
combine? Yes. So 4 into 4, you will get 16, right? So 16
Yeah.
possible conditions are there. So if 16 possible conditions are there, but still you have 20 amino acid, still there is a deficiency of 4. So
Mm-hmm.
16 code for 16 amino acid. So how other 4 amino acid will be coded by? Like coded by what? Coded by what type of code? Because only 16 codes are there, and 16 codes can code only for 16 amino acid. So what about these four amino acid, the leftover ones? Rest
Food.
of the four, where will they go? So
Food.
now, so that's why this is rejected. This is also rejected because of 4. So now it has seen that maybe they are present as a triplet like
Oh.
this: AUG, like
Oh.
GUG, like CUG. So they are present as triplets. So if they are present as triplets, so how many codons are there? Can you guess? 64. 4 into 4 into 4. So it means
Oh, four power.
we so we have total 64 codes. See, code can be more, but it
Hmm.
shouldn't be less than amino acid. So it
Okay.
is finally accepted. So it means 64 codes can code for the 20 amino acid. Okay,
Hmm.
it is something like the like for the basic amenities, for getting the basic amenities. Suppose your monthly budget is if I talk about it in Indian rupees, your 10,000. So suppose
Mm-hmm.
if your salary is 50,000, suppose, okay, and the basic amenity cost is 10,000. So if you are getting some 40 extra, so it is good na. That's why if you are getting extra code to code, so it's good. But suppose if you are getting 6,000 rupees only monthly, and your
4,000.
basic amenity cost is 10,000, so is it enough?
No.
So that is the same thing here. You can't form codes less than the amino acid. Yes, you can form more than amino acid. So that's why we choose. So we finally we came on this conclusion: genetic code is triplet. It is not doublet. It is not present as single wise. So they are present as like 3 codes together, which are called the triplet. Got it, everybody?
Yep.
So now can I say it is a triplet codon?
Mm-hmm.
Triplet. So codon is triplet. Triplet codon it is. Triplet one. Fine.
Yep.
So now, so yes. Okay. On this, we will find something. Yes, 20 amino acid is coded by these 64 codons. So definitely some codons are those, like suppose if I have AUG and if I have CUG, so they can code for same amino acid na,
Mm-hmm.
because we have so many codes. So if we have, because only 20 amino acids are there and it is 64, so definitely some codes are there which can code for same amino acid. Am I right?
Mm-hmm. Yep.
Okay. So now there was like Indian scientist, you have to remember that for the purpose
Oh
of
yeah.
NEET examination, Hargovind Khurana.
Who?
Hargovind Khurana. He
Are.
worked really well in the field of genetics. So Hargovind Khurana. So Hargovind Khurana. So he synthesized RNA molecule of definite combination. Like with the help of the DNA, he synthesized a RNA molecule. And his like with a definite combination. So he wanted some RNA molecule with a definite pattern, and he made that. So RNA molecule with a definite combination of bases.
So how to go in? Oh, Govind. Okay.
Khurana, Har Govind, like Sikh, Punjab. Do you know Punjab?
Yeah. Yeah. Yeah.
So Har Govind Khurana, definite
Kurana.
combination of bases.
Kurana. So RNA synthesis, right?
Okay, so yes, so he synthesized of course, from the DNA there is a RNA formation, right? So he synthesized the RNA molecule from the so many bases, okay, on the DNA. So Hargobind Khurana was very famous on that. Okay, so there was another scientist whose name was Marshall Nierenberg. Marshall Nierenberg.
Um, Sounds American.
Marshall Nierenberg. So Marshall Nierenberg, so he also made a cell-free system. I mean, he formed protein synthesis. Okay, like for reading the code, for reading the code. So he made a system. So it is better to say that he made a cell-free system. Cell-free system. You just need to remember only these lines because you don't have to study in very much detail all these scientists, but you should know the names because these are very familiar, very famous scientists in the field of genetics. So cell-free system
Marshall
for protein synthesis, and that
Nierenberg
help in reading the code, or you can say deciphering the code. That help in deciphering the code.
cell.
Okay. So now I am going to show you. Suppose if I'm saying that like there is a this code. So this code is valid or not? Is it triplet or not? This code is triplet or not?
Yes.
Yes, it is cytosine, uracil, cytosine. So yes, it is coding for the leucine. Leucine is a basic amino acid. So when there is a mRNA and on the mRNA there is a this code, so it will form the leucine. Suppose if there is a AUU, or suppose if there is a if I say ACU, ACU. So if there is a ACU, it is coding for threonine. Okay, threonine. So these are the amino acids. So many amino. Like if I say CGU, so it is arginine. These are the aminos. Like there are so many amino acids in our body, like phenylalanine, leucine, serine, tryptophan, glutamic acid, aspartic acid, tryptophan. Okay, phenylalanine and aspartic acid, alanine, valine, glycine, serine. So many. Proline, leucine. Okay, so arginine. Same we have like CGC, it is also arginine. Because I told you, because you have 64 codon. So many codons are there which can code for same amino acid. Same. You also have CGG. It is a codon and it is also forming same amino acid. So you will find so many amino acid which can code for same thing. Like if I say CCU, if I say CCC, if I say CCA, so all these code for the proline. So it means we have so many amino acids. No, we have so many codes which can code for the same amino acid. So it means your protein chain. See Vishnu. Suppose Vishnu, there is a mRNA and there are so many ATGC, ATC, and like code is there. So first amino acid, then second amino acid, then third amino. Yes. See Vishnu, we have the in your body. Suppose there is a one code. Suppose this is your code. AUC, CUC. I am saying CGU, ACU. So just tell me, this codon will make which amino acid in your body? ACU. Can I say threonine? Threonine. CGU. It is forming arginine. So it means so many amino acids are together combining and it is forming the protein. That's why your dad and your body has similar kind of shape and the design, because you people have the same protein, because you people have the same type of codes. So if you have the codes on the DNA, then you will get almost similar complementary code on the mRNA. And from the mRNA, the sequence of amino acid, first proline, second arginine, then glutamic acid, then isoleucine, such type of, it is also same like your dad. That's why your face looks similar. Okay. If I change the code, If I change the order of amino acid in your body, then you we will not look like your father and mom, mom and dad. Why? Because I have changed the sequence of your amino acid. And to change the sequence of amino acid, I have to change the order of this codes on mRNA. And if I want to change the order, I have to change your DNA. That is what happens in the mutation, isn't it? If you change a
Yeah.
small DNA, then change mRNA,
Mhm.
then change mRNA, and there is a different code of amino acid, different code of amino acid, then there is a different protein.
Mhm.
Got it
But
now?
I don't think you can do that though, right?
What?
Is it physically possible to like
No,
mutate
no, no,
someone?
no, no. We can't change that for the human
Okay.
beings. Okay. So now I am going to show you the genetic code. Okay. So that you can realize like how can we make so many, so many. See, it is better to show you this diagram. Can you just see that? This one. Can you see that on the right side?
Yeah.
See,
Mhm.
actually this is written as a glycine, Gly glycine. So glycine is formed by take G U GGC GGA. So all these four help in the same amino acid, right?
Okay.
So if I want to say valine, so can you tell me how many codes are available for the valine? How many codes? How many codes are there for the valine amino acid? And we say,
Uh,
see. One,
five.
two, three, four. Wait one second. One, two, three. Only four, na? See, G
Four.
U G,
Okay.
G U A, G U C, and G U U. Only four.
Oh, so GU doesn't form Valine.
N- uh, I told you you have to take triplet.
What
Triplet.
what what are the 16 that can just form with two?
16. I mean, I didn't get what you said just.
Because they're like 16 amino acids that form with just two components.
No, 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 no. No. There is no existence of monoplet and duplet.
Okay.
I was just telling you how the discovery of triplet like took place. How
Okay.
they primarily think monoplate then duplet, so it was rejected completely. You don't
Okay.
have to. It is triplet now. See, okay, better to show you this diagram. Now, can you see that?
Okay. I like the radial one better, to be honest.
See. It is triplet, right? Can you see any doublet?
No.
Can you see any monoplate? No, na.
Nope.
So it is
What's
U U
stop?
U. It is U C U. Yeah, you were saying something.
What does stop mean? Like, what is stop?
Stop. Okay, I'm going to tell you. See, we have three codons, and whenever
Mhm.
the process start, see, there is a start also. Can you see any AUG is there? Yes. Can you just see AUG? Whenever there is a process of translation starts, so always translation process will be like beginning of translation is always starting from AUG. AUG
Mhm.
is always the beginning thing. And whenever the translation process want to stop, whenever there is a termination of this process, so you have to take any of these: UGA, UAG, and UAA. UGA, UAG, and UAA. So you have to take all these three to stop the process of the translation. Okay.
They're
So
like
it
Terminator.
means, so whenever there is a translation beginning of the process of the translation, so it is started by the AUG but terminated by the three codons, and these three codon can't code for any amino acid. Their function is just to stop the process of translation. Okay.
Okay.
That's why these three codon also known as nonsense codon.
Mhm.
Nonsense means like they are not coding for anything, they are just stopping the process of translation. But the other ones, they are coding for the different type of amino acids.
Mhm.
Okay, are you sure?
Yep.
So now, yes. So yes, I was showing you the, okay, so there is a some yes. So these codons, like triplet, they are present on the DNA or RNA, mRNA? Of
mRNA.
course, on both. I mean, DNA also have
Okay,
some
yeah.
code, na? There is no uracil in that. But yes, in the mRNA, there is a presence of these.
Okay.
Okay, so now I am taking the silent features of the genetic code. It is very important. So yes, silent feature. Yes. So
What are silent
codon
features?
is monoplet, doublet, triplet, quadruplet. I mean what? Yeah, tetrate, whatever. What should I say? Codon are what? Mono, di, tri, tetra, penta, what? Codons.
Try.
Yes. So this is the first thing. Codons are triplet.
Three.
You have to note it down. Codons are the triplets. Okay. How many total codons are there? Total. How many codons? Total. How many total code
Hold
are
on.
available?
I think my internet connection is on. My internet connection is unstable, sir.
Yes. So it is 64 codons, right? There are total 64 codons. I told you. Codon means
Hmm.
what? That codon which code for the amino acid together. Codon. Okay. So out of 64 codon, can you tell me how many are nonsense codon? You just saw?
Three.
Yes, three are the nonsense codon, right? Why
Mm-hmm.
are they called nonsense codon?
Because they stop the process of translation.
Very good, because these are the stop codon. Can I tell the name? UAA,
Oh, UAA,
UAA,
UAT,
UAG, and UGA,
UAT.
right?
Oh yeah,
So
yeah.
these are the nonsense codon. You have to remember. So rest of the 63 are sense codon.
Mm-hmm.
Why? Because they code for the good amino acid, right? Okay. See. There is a line. Listen very carefully. Each codon code for one amino acid or it can code for two amino acid. Each codon, a single codon.
More than one, I think.
No.
Why not?
A single codon code for one amino acid only, na? A
Oh
single
yeah, yeah,
codon.
yeah. Okay. Yeah. Yes.
See. This is UUU. So
Yes. Yeah.
UUU is coding phenyl, okay, phenylalanine. So UU is also coding for another one, any other amino acid.
No. Okay.
No,
I
no, no.
I understood it incorrectly.
Okay,
I think.
so one codon can code only for one amino acid. So this property is called the unambiguous. Okay, so can we say that
Okay.
each codon, I got your point. I will come to you.
Mhm.
Yeah, I'm coming on that point
Yeah.
also, which you are thinking. Each codon codes for only one amino acid, only one amino acid, or you can say particular one amino acid. And this process is called, so this is known as the unambiguous. Unambiguous.
Unambiguous.
Yes, unambiguous. But you know it very well, one thing. But if I talk about the amino acid, so amino acid is coded by only one codon, or it can be coded by more than one codon. Amino acid. Hello?
More than one codon.
Yeah. Can you just see that? Oh
Hello.
my God. Wait a second.
More than one codon.
Yes. Can you just see? Suppose this is a
Answer
leucine.
me.
Can you just see? L-E-E-U.
Mm-hmm.
So this is
Yeah.
a leucine. So
Mm-hmm.
leucine is coded by one codon, or there are four codon.
Four.
So it means amino acid can be coded by more than one codon, na?
Mm-hmm.
So this property is called degenerate. Okay. So you better to say that. So but some amino acid
Only have one though.
are coded by coded by
More than one.
more than one codon. This is called degenerate. So this
Hmm.
property is called degenerate. Okay. So these are the two, unambiguous, and you can also say specific one. This is called specificity. Specific. Got it now? Yes.
Mm-hmm.
Okay, so you are reading the code with
Mm-hmm.
punctuation or
Yep.
no punctuation? Like you just read AUG. So you are reading AUG or you are reading like a comma U comma G? No comma are there. Okay, so there is
AUG.
no punctuation in it. Okay, so there is no commas, there is nothing. Okay, you don't have to read that. And remember, this genetic code is universal. Like if AUG is coding for methionine amino acid, so in all the organism, in all the animals, in all the plants, AUG is coding for the methionine. AUG is coding for methionine in all the organisms. So it means you are reading the genetic code. So if you are reading the genetic code, so it is only for yours or it is also for the other ones, all the living
Other
organism.
ones.
So that's why you are calling it as genetic code is universal.
Mm.
Got it now?
Yep.
And AUG it is last. AUG. AUG is what I told you. AUG has some specific function. AUG. Think. AUG
Oh,
has
it
some.
starts the thing. It starts
Yeah.
translation.
So it is. Can I say it is known as an initiator codon? Initiation. And
Yeah, yeah, yeah.
not only initiation, it is also coding like stop codon is stop codon. It is not coding for amino acid, but it is also only for initiation or it is coding for something. Coding for something. So that's why it has a dual
I think
function.
it's like methamine.
Methionine
Methamine.
amino acid and code for.
Oh,
So it
methionine.
means if it is the starting the process, so it means whenever there is a protein chain in human being, so it is always starting from the methionine because it is starting initiation.
Mm-hmm.
Vishnu, because AUG is a dual,
Yeah.
it is a starting codon now, so of course it will form the methionine first at the first number. Rest can be different, but first remain the same methionine. Are you
Sorry,
getting my?
sir, my
Yeah.
bandwidth is low. Like my network is very bad today.
So now we are going to start a very important process, translation. Translation. Okay. You have seen that from the from the DNA we have formed the mRNA chain.
Hmm.
This mRNA chain, it has some codes, yes or no code,
Yep.
and this code will basically work as a codon. Like suppose
Hmm.
this is CGAUGGCGA, something like this, so on. So the
Yeah.
process will start from here only, na? It is
Yes.
not starting from because AUG is the initiator codon, so it will start from here.
Yes.
This is mRNA.
Hmm.
You know it very well. So you should know we have a tRNA molecule also. I told you tRNA diagram,
Mess. Yes. Yes. Yes.
something like that.
Hmm.
Okay. So it has a reading ability also like this. Three codon.
Yes.
This is AUG and it has a reading ability for that. Okay.
Hmm.
What is happening actually? So with the help of the energy, amino acid, like whenever you eat a lot of food, so in your body there is a amino acid, right?
Hmm.
From the protein you are getting the amino acid, na? Lots of amino acids. Tell me.
Yes.
So with the help of the ATP, energy utilization, the amino acid are utilizing your body energy and it is going to combine with that portion. That portion. Okay. So initially tRNA was non-functional, but now tRNA is completely functional. Suppose there is a protein which is there is a amino acid which is known as methionine. So now
AUG.
so methionine is going to attach here. So now
Yes.
now this methionine need which code? This
AUG.
methionine yes. So this is coming over here now.
Ah.
So when this will come here, so it will make a anticode. This is AUG. So A is connected with A is connected with
Uh,
U is connected with A,
U.
and G is connected with C. So UAC, it is
Oh.
connected that, right?
Oh, it's like the opposite.
Yeah, anticodon, right? So
Oh.
now it has come over here like this, and now
Okay.
it has bringing the amino acid. Now fine. Now next is CGC. So there is another tRNA, lots of tRNA in our body. So suppose,
It forms the anti-code for that, which
yeah,
is
suppose there is a
GCG.
so yes, suppose there is another protein, another amino acid like histidine. Suppose histidine is
Okay,
there.
okay.
So this CGC is coming over here,
GCG.
right? Yes, GCG.
GCG.
So it is coming over here, right?
Yes.
Something like this. So it is bringing the amino acid, yes or no?
Mm-hmm.
This is amino
Histidine.
acid 1, this is amino acid 2, right? Suppose, so just tell me, whenever this thing happen, na, so there is a, I told you rRNA process, remember? Burger-like structure, remember
Oh
that?
yeah.
Which is
Yes,
catalyzing the
yes.
process. Without
Mm-hmm.
rRNA, it is not going to happen. You have to put
Okay.
that. So now just see, this tRNA and amino acid bond is not so strong. Okay, what I just said, the amino acid and tRNA bond is not so strong. This amino acid and tRNA is also not so strong. So this amino acid, can you just see that? This
Yeah.
amino acid is leaving the tRNA and it is coming over here. Okay, this is
Mm-hmm.
coming over here on this amino acid. So now it become a chain of two amino acid, yes or no?
Yes.
Because they are not so stable, but amino they are stable. Peptide bond, two
Mm-hmm.
amino acid. Okay. Now there is a next tRNA, isn't it? Same thing is happening here again and again. Next tRNA is
Mm-hmm.
coming like that. This is the third amino acid. Okay. This is amino acid 1, 2, this is 3. So this 2 is coming over this 3. Okay.
Yes.
So now it will become a chain of how many amino acid?
3.
3. So this will continue so forth and so on. This rRNA is going along with that. And finally, when the stop codon will come, suppose UAA. So this process will stop or not?
Say it again.
UAA, stop codon.
Oh
Remember,
yeah, yeah.
remember.
So it's gonna stop. Yeah.
Yeah, so this process is going to be ended here. Yes, so now I am showing you a video. Okay, this video will help you in understanding the translation process uh, very easily. Yeah, so see this video.
Translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formal
This is
methionine
rRNA, I told
tRNA,
you,
and messenger RNA.
rRNA.
The 50S ribosomal subunit.
Can you just see? This is AUG, this is UAC. It is bringing some amino
Yes.
acid. Can you see methionine, MET, methionine? So ribosomes are actually in the two unit burger-like structure. Na? Burger are two structures. So this is one
Yes.
structure of burger and this is the other structure of burger, the pink one. And they are catalyzing
Okay.
the process.
Unit then joins the complex.
See, it
Proteins
look like
called
burger, right now? I know a little bit something like this. So this pink,
Yeah. Yep.
it is completely a
Yes.
burger. It is bringing
Okay.
amino acid and it is coming over that coding.
Okay.
initiation factors are also involved but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A.
Remember, so this is the first tRNA come here. So the next tRNA will come A side or the back side of P side. Next tRNA. Next tRNA will come here, na A side. Yeah, acceptor. A means acceptor site.
A site. There is also a third site called the exit or E site, where transfer RNAs are released. The initiating transfer RNA
This is
I
the
see.
exit side actually. Whenever the stop
So-
codon will come,
Mm.
you will see.
carrying formal methionine binds to the P site.
This is the next
A transfer
tRNA.
RNA that
Can you just see that?
recognizes the
It is
next
coming
out-
codon
coming
and
with the
carries the
protein.
second amino acid, then moves into the A site. The formal methionine carried by the transfer RNA
But these tRNA and amino acid, they are strong or weak?
Strong.
No. Amino acid and tRNA.
Oh, weak.
So this amino acid is coming over this, right?
Peptide bond is
Yeah.
strong, right?
In the P site
Can you just
is
see
then
that?
joined to the amino acid
Yes.
carried by the transfer RNA
Now
that
the
just
next will
entered the
now
A
it will be
site by a
removed.
peptide
Now this tRNA will be removed, which is at the P site.
It's done. It's
Yeah.
bond.
done.
The ribosome now advances a distance of 1 codon,
This
and the
is catalyzed
transfer
by
RNA
this process is catalyzed by the rRNA. Can you just see that the burger-like structure?
Oh.
It is catalyzing the
that
process.
carry the
Yes.
formal methionine is released at the E site.
Now exit side.
transfer-
It is becoming exit side, right?
Oh.
So it is going to leave something
Yes.
exit side.
I understand.
RNA carrying the
So
next
now new
amino
tRNA
acid
comes in.
now moves into the A
Yes.
site where the
Tyrosine.
anticodon on the transfer RNA
Yes.
matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site.
Can you just see that? Now
The ribosome
amino.
continues.
Now the protein is formed by the 3 amino acid, and same thing will continue, so forth and so on, right? It
What
keeps
is
on continuing.
FMET and Pro?
What is?
What is FMET and Pro?
No. You have to see just this thing. Methionine. First. F means
Oh.
first. First.
Oh, what's Pro?
Proline.
Oh,
These
I don't
are
know
the
the name
okay.
of the
These are the
peptide.
name of amino acid. 20 amino acid, na? We
And
have
TYR is
20. We
tyrosine.
have 20 amino acid: tyrosine, threonine, methionine, aspartic acid, glutamic acid, serine, histidine, tryptophan. So many amino acid. Okay.
Okay.
To
Would it
move
be
along
helpful if
the
I studied
messenger
all of them?
RNA, and new amino
Aspartic
acids are
acid,
added to the growing polypeptide
proline
chain.
again,
Proline.
tyrosine again,
Elongation of the
aspartic acid.
polypeptide is terminated
Oh,
when a
acid.
stop codon moves into the A
Now,
site.
can you just see that? Stop codon is coming.
UAA.
Yeah, if UA is coming, so process is going to stop or not?
Stop.
A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and
And then
50S
finally everything will be removed, right? And a chain is formed in your
Yeah.
body. Can you just see that?
subunits.
Finally,
The messenger
the-
RNA and
everything is broken, and finally the chain will be formed. So this is what we want, right? The chain is already formed. We want this chain
Yes.
only, na, protein. So this is known as what? This is known as polypeptide chain. Am I right?
Oh,
So this is
polypeptide. I see.
polypeptide means there are so many peptide bond
Peptide bonds and
yeah,
proteins are formed by peptide bonds.
yeah, and it is completely formed. What completely formed? The protein chain, right?
Protein chain.
So this is what we wanted, right?
Yeah.
So we have completed the transcription, and now we have also completed the translation process. Am I right,
So
everybody?
proteins are formed by DNA.
Yes. No, you are eating a food. Amino acid is not coming by DNA. Amino
No.
acid you have to take by your food. DNA
Yeah,
is
but
only making in sequence wise. Sequencing. DNA is
So
only helping in sequencing of these amino acid to form the
yeah.
protein.
So
Okay.
I took a cooking class in like last year. So I heard that like you can only make so many amino acids in your body, but the rest of them you have to eat.
Yeah,
Right.
actually there are two type of amino acids, okay? Essential one and non-essential one. So some amino acid your body can prepare, but some
Mhm.
are very essential one your body can't prepare. So you have to take it from outside. So
Okay.
essential and non-essential. Non-essential, there is no need to take from outside because your body can easily prepare them. But some are very important, like
How many of
methionine.
each are there?
How many of each? It is
Yeah.
approximate essential one are approximate 8 to 9, which I remember. 8 to 9 we have to
Okay.
take. Okay, so
But
some amino
total amino
acid
acids are 20, right?
yeah, 20 which your body
Okay,
need.
okay.
Okay,
Yeah.
so I told you something in the translation process. So amino acid become joined by the peptide bond, right, to form the polypeptide chain. So
Yes.
firstly, amino acid are completely free right now. Okay, just I want to join with tRNA. So amino acid are completely free. So
Uh-huh.
amino acid are taking the energy of your body, ATP.
ATP.
I told you. Okay. So by using the ATP, now it is making a complex. Complex
Oh,
means
okay.
amino acid. Okay. And ATP is broken down into the
ADP.
adenosine monophosphate.
Oh, AMP. Oh,
Monophosphate. An
that's
enzyme.
diphosphate.
Yeah. And two phosphate are removed. Two phosphate are
Okay.
removed. Okay. It is
Where do they go?
monophosphate. They will remain like there only. Okay. You don't have to take care of that. Okay. So it is a
Okay.
complex now, right? Now now this complex is attacking on tRNA. You remember na, that cross-like structure. So now this
Oh yeah,
tRNA
yeah,
is
yeah.
completely free, and now they are going to attach. And now when they are coming with that, okay, so then what will happen? So now then amino acid is going to attach on that, yes or no? Now
Yes.
tRNA is free here, and amino acid is also free here. But now
Mm-hmm.
after this reaction, amino acid will be attached to tRNA. Fine. Initially, amino acid is free here, and tRNA is also free here. But after the
Mm-hmm.
reaction of that, they are not free here. Okay. So now so now can we say it is
Mm.
a activation of tRNA? Activation of amino
Yeah.
acid, because the amino acid is going to attach with what? Initially, amino acid is this, tRNA is this. So amino acid taking the ATP, it is becoming a complex. And this complex is combining with tRNA, and then amino acid is going to attach. And rest of the portion will be removed. Right?
Yeah.
Like AMP will be removed. That single phosphate is also removed. Okay, everything is utilized. And now this is the what we want. In the process we just saw na, they will come one by one at the respective code.
Yeah,
Yes
and
or
all
no?
the things at the top would remain
Yeah. So
connected.
now it will code here, and the respective amino acid will come. Got it? In the
Yep.
AUG, can other amino acid come? No.
Hmm?
The code will not match na. Every amino acid has some code na. So that will not match. Like you just saw AUG, methionine has a code of UAC. So can this come? GCA?
No. It has
No,
the exact
because
opposite.
yeah, it is not matching with that. So can it come first? No, no.
It has to be the exact
Well,
opposite of
yeah.
what it's on the codon thing.
Okay, so if I talk about the initiation process, so Vishnu, what you have just saw in the video, rRNA were coming, right?
AUG.
AUG is there, but rRNA is also there, na? rRNA has how many site? That
Oh
pink, pink,
yes, the
pink, pink.
catalyst, the burger,
Yeah, yeah. So it has how many
burger.
site? I mean, how many subunit? One, two,
Two,
three, four. Yeah,
50s and 50s
yeah,
and 70s.
yes. No, 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 no. 30 and 50. Okay,
Oh, 30 and 50.
yeah. So large
Oh, I
subunit,
think I heard 70 somewhere.
it
I
is
know I heard.
total 70. 50 plus 30 becomes
Oh,
70.
okay.
Okay, it is a different mathematics here.
That's how math works.
Okay,
That's
so
how math
large
works.
subunit, yeah, it is basically on the like like sedimentation coefficient, how much heavier it is. So it
Oh,
is not that mathematics. Okay, so large subunit and small subunit. So this is I mean the RNA is coming. Which RNA? rRNA. So when this rRNA is coming over
burger.
what? mRNA, am I right? Like this.
Mhm.
So now the site is becoming active or inactive?
Active.
Now the translation will begin, right? Still there is no
Yes.
translation. But when they are attaching to the site, now translation will begin. So can we say that when
Hmm.
the small subunit encounter small subunit, firstly small subunit will come. When the first small subunit will come, so translation is going to start from here. Okay, and now the mRNA will bind to the small subunit. Okay, and then following base pair will happen. What? Some initiation factor. Okay, so now after that, you know it very well, the large subunit is also there. So how many sites are there? You have seen that, how many site?
Two, three, three, three.
P site, E
A site
site,
and E site.
A site, A,
Exit
A.
site, accept site, and P site.
Yes. So firstly, if I talk about first tRNA, it has methionine. So it will come at E, P or A.
P. No,
No,
A.
no, no, no.
Oh,
Firstly, beginning.
okay, firstly, then P, and then A,
Uh,
and then it keeps on going.
yeah,
Yeah.
yeah, yeah. Firstly, it will come at P, and then the next one will always come at A, and it keeps on, and then it is going to eliminate it when it form the peptide bond, right?
Yeah.
Am I right? So this is
Yeah.
how this process keeps on continue. Okay. Any doubt in this?
Nope.
So so this is the initiation. There is a process with like there is a process. It will look something like this. You have to remember this diagram. Elongation of what? Polypeptide.
Yes.
So I want to show you in the picture so that you can see that. Just give me 1 minute. Like this diagram will look like something like this. Yes, can you see this diagram in your
Mm-hmm.
copy? 1 second, just I can show you the. Now can you see that? This
Yes.
diagram. So this is large subunit, this is small subunit. This tRNA is going to be removed, right? And this is coming,
Mm-hmm.
and this is at the P site. Can you just see that? So you
Right.
have to draw this diagram in your copy, okay? Please note it down. You can click a picture, or you just remember
Okay.
this is known as the elongation. Okay, this is the elongation of polypeptide. You have to remember that. Okay, and can you just see this is U. Can you just see triple U? Like UA, whenever there is a, like, can you just see UAA? UAA, yeah. Whenever there is a stop codon, so this process is going to be stopped. Whenever there is a stop codon, so this
mRNA is very
process,
important in both of these.
what?
R rRNA.
What you said? What you said?
Okay. Oh, I said messenger RNA is important in translation transcription.
mRNA is coming out from the transcription,
Oh,
and
I'm saying
now,
it's important.
yeah, and this mRNA, yes, because everything is happening on the mRNA. Everything is happening on mRNA, no? Because this mRNA has a code of DNA, which is coming out from DNA, right?
Mm-hmm.
From the DNA, this is taking out the information, and now this is the mRNA on which everything is happening. Got it?
Yep. Yep.
So mRNA is very important. Other things are helping only. Okay, got it now?
Mm-hmm.
So here, okay, if I ask you how the termination of polypeptide will take place, initiation,
I
elongation
know.
diagram, you have seen that, and termination
Yep.
will happen
Uh,
how?
until one of the stop codons occur in the mRNA.
Yeah, and how many types of stop codons are there?
3.
Yes, can you tell me the name?
UAA, UAC, no, UGA, UAG.
Okay, remember in the initiation there are some initiation factor, and in the termination there are certain termination factor too. Same like in the transcription, remember sigma and rho.
Sigma and rho.
Yeah, so same something like this. So you have to remember just a initiation factor and the termination factor. Okay.
Mm-hmm.
So today we have covered this genetic code and the translation process. Okay,
Mm-hmm.
Without diagram you won't get marks. Yes, but in the NEET examination you have to remember the concept. Okay,
Okay.
so we are done with that here today.